Hello everyone, welcome back to our next class, Non-Chordates, Unit 1 Protista. Today I will be discuss about study of amoeba. Let's come to know what is amoeba. Amoeba is a unicellular organism that has the ability to sense its shape. They are usually found in water bodies such as ponds, lakes, slow-moving rivers. Sometimes these unicellular organisms can also make their way inside the human body and cause various illnesses. The term amoeba comes from Greek amoebe, meaning to sense. This may be due to its shape that is constantly sensing. Variants amoeba. One of the first reports referencing amoebas deaths can be traced back to 18th century. A German naturalist named August Johann Rosenhoff, who discovered a specimen and made detailed illustrations of the same in 1755. So this is the history of amoeba. See the classification of amoeba. Traditionally, amoebas are grouped under the kingdom Protista as they are neither classified as a plant, animal, nor fungus. However, amoebas fall under the eukaryotes as they possess a true nucleus. Amoeba is a genus of single-celled amoeboid in the family Amoebidae. Here in the chart you see kingdom Protista, phylum Protozoa, class Sarcodina, subclass Rhizopoda, order Amoebida, family Amoebidae, genus Amoeba, and species most common Proteus, Animalculae, Dubia. These are also another species of genus Amoeba. Let's come to know the structure of amoeba. Typically, most amoebas are characterized by the following features. Movement occurs through the use of pseudopodia, where the cytoplasm pushes the plasma membrane outward or inward, creating blunt, finger-like projections. I think you all are aware about the pseudopodia or the finger-like projection body of amoeba. Number two, there can be multiple pseudopodia at one particular instance, hence each shape rapidly changes. Structure of amoeba primarily encompasses three parts: the cytoplasm, plasma membrane, and the nucleus. The cytoplasm can be differentiated into two layers, the outer ectoplasm and the inner endoplasm. The plasma membrane is a very thin, double-layered membrane composed of protein and lipid molecules. Amoeba also contains other cellular organelles such as a contractile vacuole, mitochondria, Golgi apparatus, and fat globules. The major function of contractile vacuole in amoeba is for osmoregulation. It regulates osmotic pressures within the cell. It does so by filling itself with excess cytoplasmic water known as diastole. Then it fuses with the cell membrane to release the content outside by a quick contraction known as systole. Amoeba consume food either through the process of phagocytosis or pinocytosis. I will be explain next the feeding of phagocytosis. The process of reproduction is through asexual means such as binary fission. The lifespan of a typical amoeba is 2 days, but because it undergoes binary fission. The resultant daughter cells are the same as its parent cell. So technically amoebas can be termed as immortal. When living conditions are not ideal, an amoeba can essentially transform itself into a protective ball called a microbial cyst. When living conditions become better, it can revert back to its trophozoite stage where it can start feeding again. Here in the figure you see the pseudopods, finger-like projection, food vacuoles, nucleus, outer membrane. This is ectoplasm, endoplasm inside, and it is contractile vacuole. Let's come to know about the characteristics of amoeba. Amoeba, a unicellular organism found in stagnant water. The size of amoeba is measuring about 250 to 750 μm or micrometer. They move with the help of finger-like projection called pseudopodia. Cytoplasm is differentiated into two parts. Outer portion is ectoplasm and inner portion is called endoplasm. Amoeba are eukaryotes whose bodies most often consist of a single cell. The cells of amoeba, like those of outer eukaryotes, possess certain characteristic features. Their cytoplasm and cellular contents are enclosed within a cell membrane. Their DNA is packaged into a central cellular compartment called the nucleus. Each amoeba contains a small mass of jelly-like cytoplasm, which is differentiated into a thin outer plasma membrane, a layer of stiff clear ectoplasm just within the plasma membrane, and a central granular endoplasm. The endoplasm contains food vacuoles, a granular nucleus, and a clear contractile vacuole. People usually become infected from warm freshwater lakes and rivers. Here you see the feeding habit or phagocytosis of amoeba. The amoeba cell surrounds its food. For example, bacteria, other protists, detritus, which eats pseudopods. When food comes in contact with an amoeba cell, the ectoplasm forms a tube called ectoplasmic tube, takes the food into it, and then converts it into a food vacuole. The eating process of the amoeba can be likened to a human phagocyte executing phagocytosis of a foreign body. So this term is familiar for you because this term is used in case of blood platelets or blood cells, which shows this amoebae nature or phagocytosis nature, which engulf the foreign particles or disease particles inside the body. Now you see the nucleus of amoeba in this figure. Food particles come in contact, the pseudopodia come out, capture the food, and then food vacuole forms here, and thus it's then engulf the particles. Here see the term food when come intact, pseudopodia capture the food, that is happen ingestion, and then digestion occurs, and then absorption takes place, and then assimilation happen, and finally egestion takes place. So this is the mechanism of food habit or the feeding habit or phagocytosis. Now you see the amoeba movement. As a class of organism, amoeba are defined by their unique movement patterns. This movement strategy produces forward movement via the following three steps. Number 1. Ballooning the plasma membrane forward. This distinct rearrangement is known as a pseudopodium or false fold, which is very similar in nature to that of the lamellipodium generated in higher vertebrates. Number 2. The pseudopodium attaches to the substrate and is filled with cytosol. Number 3. The rear portion of the amoeba releases its attachment to the substrate and is propelled forward. So this mechanism is happen during the amoeba movement. During amoebic movement, the viscosity of the cytosol cycles between a fluid-like sol which flows from the central region to the cytoplasm known as the endoplasm into the pseudopodium at the front of the cell. Once this occurs, the endoplasm becomes an ectoplasm containing gel-like substance that forms the cortex under the plasma membrane. As the amoeba moves forward, ectoplasmic gel is converted once again into the endoplasmic sol, and the cycle is repeated as the cell continues to move. This transition between the gel and sol states occur following the collapse and reassembly of networks of actin microfilaments located in the cytosol. In particular, coffinin is responsible for the disassembly of the actin filament to form the sol, whereas profilin leads to actin polymerization and the gel is formed by alpha-actinin and filamin. So this is the chemistry behind it. Let's come to know about the amoeba reproduction. Due to the extremely diverse natures of amoeba, the various species of amoeba reproduce using a variety of different methods. These methods include spores, binary fissions, and even sexually. Let's see binary fission. By far the most common form of asexual reproduction employed by amoeba is binary fissions. In preparation for reproduction, the amoeba will withdraw its pseudopodia and form a spherical shape. Mitosis is observed in the nucleus and the cytoplasm divides at the center of the cell and separates, forming two daughter cells. Since this process involves simply copying the genetic information to form a second cell, the two resulting daughter cells are identical clones of the parent cell. Thus, the nucleus is absolutely essential for this form of reproduction. This has been verified in experiments involving slicing an amoeba in half or extracting the nucleus from the amoeba. In both situations, the cell eventually dies without a nucleus. Multiple fissions and encysment. Under conditions of food shortage amoeba will reproduce via multiple fissions. This process involves the production of multiple daughter cells by the pseudopodia being retracted and the amoeba forming a spherical shape. Secondly, the amoeba secreting a substance that hardens and encapsulates the cell, forming a cyst which is called encystment. The amoeba, protected by the cyst, will undergo mitosis several times, producing multiple daughter cells. When favorable conditions return, the cyst will burst, releasing the daughter cells. Within a host, the amoeba will undergo encystment as a means of protection against desiccation as it travels through the colon, which ensures its survival outside of the host. Here, the figure you see binary fission and multiple fissions. The fully grown amoeba, when it follows the binary fissions, the amoeba retracts its pseudopodia and the nucleus divides by mitosis. Daughter nuclei separates and cytoplasm constricts. Then finally, two daughter amoeba cells produce. When it follows the multiple fissions, same amoeba pulls its pseudopodia. Then the amoeba secretes a outer cyst wall forming. Then it's the amoeba causes inside the cyst mitotic division of amoebic cells, and under favorable conditions, the cyst ruptures and releases many daughter amoebas. So in such a way, amoeba reproduce following the binary fissions and multiple fissions. Now you see the spore formation. Solitary haploid amoebae known as the myxoamoebae or social amoebae, this is the new term, resides on decaying vegetations, that is logs, eat bacteria and reproduce asexually via binary fissions as described above. However, unlike the amoeba, which undergo encystment when the food supply becomes exhausted, tens of thousands of myxoamoeba will fuse, forming a moving stream of cells converging at a central location. It is at this region that the cells stuck on top of the other and form a conical mound termed a tight aggregate. Next, a tip rises from the top of the conical mound and the tight aggregate folds to produce a mobile grex, also termed a pseudoplasmodium or slab. It is 2 to 4 mm in length and surrounded by a slimy substance. The grex will then migrate towards the illuminated area where it will differentiate into a fruiting body composed of a tube stalk, which is approximately 15% to 20% of the entire cellular populations, and spore cells. This process involves the secretion of an extracellular coating and the extension of a tube through the Greeks by pre-stalk cells located in the anterior of the Greeks. As the pre-stalk cells differentiate into stalk cells, they create vacuoles and become enlarged. This serves to lead the pre-spore cells in the posterior section of the Greeks. The elevated pre-spore cells differentiate into spore cells and disperse, each representing a new mesoamoeba while the stalk cells die. Now you see the sexual reproduction. Mesoamoeba are also unique in that they can also reproduce sexually. This occurs when two mesoamoeba fuse to create a giant cell. This giant cell will then engulf all other cells in a mesoamoeba aggregate. After ingesting all of its neighbors, the giant cell will encase itself and undergo meiosis and mitosis divisions a number of times under the protective cover of the cyst. Means both happen, meiosis and mitosis. When appropriate environmental conditions are met, the cyst will burst, releasing new mesoamoeba. Since this process involves meiosis and the genetic information from two amoeba, the resulting daughter cells will be genetically distinct from the parent cells. So this is the mechanism of sexual reproductions, which the mesoamoeba come from the spore formations, derived from spore formations. Here the picture you see, this is the gametogenesis happen and the gamete fusions takes place and pronuclear fusions takes place, zygote form. This is sexual aggregations of mesoamoeba. This is zygote giant cells. This too is the zygote giant cell. Endocytes form, or this is the pre-cyst. Cannibalistic phagocytosis happen. This is macrocyst form, maturation, degradation of endocyte, and then meiosis takes place, or dormancy means propagation of progenies. So in such a way, from the mesoamoeba cells, thus by forming gametes and fusions, and new amoeba cell produced through sexually. Now you see the factor responsible for the reproductions. This is temperature and reproduction. Temperature is a critical factor which impacts the amoeba growth. While several amoeba species have been found to grow at a wide range of temperature ranging from 10 degrees centigrade to 37 degrees centigrade, pathogenic strains have been found to survive more efficiently at higher temperatures between 32 degrees centigrade to 37 degrees centigrade. This indicates that amoeba are highly resistant to temperature fluctuations, and most are adapted to survival within humans. Therefore, this may have pathogenic implications as amoeboid cysts are extremely resistant to microbicides and can infect human via contaminated drinking water. So this cystis is very dangerous. It can enter human body through contaminations. Amoeba reproductions is by binary, a type of asexual reproductions incorporating mitosis and cytokinesis and sexual reproduction, that is by syngamy, seems to occur as well, particularly in certain amoebozoans. So not all species encourage the sexual. Some amoebozoans may encourage the sexual and rather it's produced by binary fissions. So this is the mechanism of and factor of amoeba reproductions. So this is the reference for your knowledge. Thank you.